Okay, good morning, dear students. Today we are going to study about recording problems and measurement with two electrodes. Okay, so while we are recording the signals like ECG, EMG, EEG, so while recording there will be some variation in the signals. So that comes under the recording problems, and the measurement with two electrode means how we are measuring this signal using this two electrodes. Okay, first just an introduction. What is electrodes? What is the purpose of electrodes? You all know. It just converts the ionic potential into electrical potential. That is ionic voltage into electronic voltage. Okay. These electrodes are mainly used for measuring electrocardiogram, electroencephalogram, and electromyogram. So the potential is generated due to the exchange of ions between metal and the electrolyte of the body. That is, the voltage is generated due to the exchange of ions. There will be ions in the body, and this will be transferred between the metal and the electrolyte. So at that, while transferring, it takes place the voltage. So the potential is generated due to the exchange of ions between metal and the electrolyte of the body. So for the measurement of the signal, a wide varieties of the electrodes are used, as you know. That is for measuring. In the last class we all learned about the types of electrodes that are used for the measurement of various signals. So what are the main three electrodes? Microelectrode, surface electrode, and needle electrode. What is microelectrode? Yes, it is used to measure the biopotential near or within a single cell. That is it acts as an intracellular electrode. That is from measurement of signal from inside or outside of the cell. Okay. Next one, surface electrode. What is surface electrodes? That is the electrodes which are used to measure the signals from the surface of the body. That is called surface electrodes. Okay, that is the measurement of voltage available from the surface of the skin, and is used to sense the potential from heart, brain, and nerves, etc. So next one, needle electrode. What is needle electrode? It is used to measure the bioelectric potential of highly localized extracellular region in brain or bioelectric potential from specific group of masses. Just it looks like a syringe. Okay, so it is mainly used in brain surgery, and also these are the three types of electrodes. Under this there will be some sub electrodes are there, so that you studied in the last video. Okay, so this figure, this is the equivalent circuit of an electrode. Okay, this is an equivalent circuit of the electrodes. Next we are going to see about the measurement with two electrodes. In the last figure we see the equivalent circuit. So in this figure, this one and this one shows two electrodes. Okay, so measurement with two electrodes. So the voltage measurement, that is the signal measurement from the body, requires two electrodes. Normally we are using two electrodes, one as the main electrode and other one as the reference electrode. So for the measurement we need two electrodes. So the voltage measured, so we know we are using two electrodes, and these two electrodes are producing some voltage. So the voltage measure, so the final voltage is the difference between the instantaneous potential of these two electrodes. So the difference between these two electrodes is called as is the main or actual voltage that we want. Okay, if we are using various types of electrodes, okay, right? So if we are using the same electrodes, that is, if the two electrodes are of same type, the difference is usually small. That is, the same electrodes will measure the potential, right? So the difference between these two potentials are very low comparing to other. That is, the difference is usually small, and it depends on the actual difference of the ionic potential between the 2 points of body from which the measurements are being taken. Okay, this is one type of one electrode, and this is the equivalent circuit of another electrode, and these two electrodes are connected to the body. That is, so here in the body also there will be some resistance, that is called resistance of the body, that is denoted here. So finally the difference between the voltages generated by the first electrode and the second electrode is called the main voltage, is the output voltage that we want after the measuring. Have you understand? Okay. So what happens if we use the two electrodes of same type? The difference is usually small, and it depends essentially on the actual difference of the ionic potential between the 2 points of body from which the measurements are being taken. Okay, then continuation. So in the last point we learned about if the two electrodes are of same type. So what happened if the two electrodes are of different type? What happened? Can anyone say what happened? Okay, that is, they produce a significant DC voltage that can cause a current flow to both the electrode as well as through the input circuit of the amplifier to which they are connected. If we are using different electrode, means there will be some current flow through both electrodes, so there will be some variation in the voltage. Okay, so the DC voltage due to the difference in electrode potential is called offset voltage of the electrode. So if we are difference in electrode potential, the DC voltage due to the difference in electrode potential. Okay, so the variation of this voltage is called as offset voltage of the electrodes. The two electrodes of same material may also produce a small electrode offset voltage. That is also we learned earlier. That is, if we use the same electrode also means there will be some low amount of voltage generated. So in that case also there will be some electrode offset voltage, small electrode offset voltage. So the chemical activity takes place within an electrode can cause voltage function to appear without any physiological input. We know in this electrode there will be some chemical reaction, right? So this chemical reaction results in the variation in the voltage without any physiological input. Such variation, so due to the chemicals inside the electrode there will be some variation of voltage. This variation results in the noise generation. Okay, so such variation may appear as noise on the bioelectric signal. So we can reduce this noise so it can be reduced, that is, this noise can be reduced by proper choice of the material or by coating the electrodes to improve the stability. So the best material for this is silver to silver chloride. So this is mainly used for this purpose. Okay, next we are just going to see a small topic under this, that is called polarization. So in the above figure we see a resistance capacitance network. Okay, so the impedance of the electrodes as a fixed value of resistance and capacitance. So on the resistance capacitor network shown in the above figure represents the impedance of electrodes as a fixed value of resistance and okay, here we know the impedance will be vary. So in the above figure we shows that the impedance of the electrodes is a fixed value of resistance and capacitance. So it will not be constant, there will be some variation in the impedance in the network also. Okay, so the impedance is the frequency depend, so it depends on the frequency because of the effect of capacitance. Both the electrode potential and the impedance are varied by the effect of by an effect called polarization. So due to this polarization, the electrode potential and the impedance will vary. Polarization is the result of direct current passing through the metal electrolyte interface. So this polarization is due to the current. We know there will be some current. It will be passed through the metal and the electrolyte interface. So because of this, due to this passage of current, the polarization will occur. So the electrodes are designed to avoid or reduce polarization. If the amplifier to which the electrodes are connected has an extremely high input impedance, the effect of polarization and change in electrode impedance is minimized. If the amplifier have high impedance means we can reduce the electrode impedance. Okay, that is we are connecting this electrodes to this amplifier. So if this amplifier have high input impedance means the effect of polarization and the change in electrode impedance is minimized. So the size and type of the electrode is important for determining the impedance. Okay, so large electrodes have low impedance and small electrodes have high impedance. These all comes under the polarization. Next, these all are the measurement with two electrodes. Then we are going to see about the recording problems that arise due to this measurement using two electrodes. Okay. The first recording problem is the inaccessibility of the variable to measurements. Okay, inaccessibility, that means we are not able to access the variable for the measurement. That is the main problem. That is, it is a greater difficulty in attempting from a living system is the problem in gaining to the variable being measured. So that is sometimes there will be some difficulty in accessing the variable for the measurement. So this is the main recording problems that is affecting. Okay, so for example, the neurochemical activity of the brain. So it is not impossible to place the transducer. What is transducer? Which converts this ionic current to electric current. So we need to do the indirect measurement. So we can't, in the neurochemical activities of brain, we are not able to place the transducer inside. So we go for the indirect measurement. By using indirect measurement, however, one must be aware of the limitation. We all know what happens if we are using this indirect measurements. So next one, variability of data. What is variability of data? From this name, from the name itself, we get there will be changing in the value of the data, variability of the data. So this value, the variables will vary with respect to time. Suppose if we are measuring some ECG at one time we get some value and another time we are getting some other value, that comes under the variability of data. So the majority of the psychological variables are non-deterministic, means it varies with respect to this time. So these must be represented by some statistical or probability distribution. Next one, lack of knowledge or interrelationship. The physiological measurement with large tolerance are often accepted by the physician because of lack of this knowledge and the resultant inability to control variation. That is, there will be some error value. That is, at this point of error we can able to it's okay for measuring. So that is the thing. So psychological measurement with large tolerance are often accepted by the physician because of lack of this knowledge and the resultant inability to control variation. Better understanding of the psychological relationship would also permit more effective use of indirect measurement as substitutes for the inaccessible measure. That is just lack of knowledge of the interrelationship. Next one is the interaction among physiological system. Okay, if for a person, if a person is not well, have many other diseases, and also a large number of equipment is connecting to this his body, so there will be some interconnection, there will be some uh, connection between the various devices, right? So at that time also there will be, that is, a large number of feedback loops involved in the major physiological system. A severe degree of interaction exists both within a given system and among the major system. So if we are just measuring a system, and if the body is already connected with some other system, means there will be some, that is just as an if you are using two equipments, the noise from ordinary equipment will affect the noise of the other, right? So similarly it will also happen interaction among physiological transfer. Then the effect of um, transducer. What is transducer? It can be considered as a device converting one form of energy to another form. What is the one form of energy? Here just we are converting ionic to electric current. So the electric transducers can be considered as the device meant to convert a form of energy to an equivalent electrical signal. The physical quantity to be measured can be position, displacement, flow, temperature, strain, velocity, etc. And the output is in the form of electrical parameters like current, capacitance, voltage, inductance, change in resistance, etc. So this when we connect this transducer to the body also, there will be some variation in the signals that comes under this recording problems. So we can see what is, let's see what is transducer. Okay, what is transducer? It converts ionic current to ionic energy into electrical energy. So it consists of two parts. This figure shows this transducer. There will be one sensing element. Then next one, transduction element. So in the sensing or sensor element, it is responsible for generating measurable response with respect to the change in physical quantity. This part is responsible for generating measurable response with respect to the change in physical. So here we get the response, for measurable response. What about the values we are going to measure? Or we know it converts this ionic voltage into electronic voltage. So that is censored by this sensing elements, what we are going to transfer. Okay. Next, transduction element. The sensor output is carried out to the transduction elements, which converts this non-electrical signal to the electrical signal in proportion to the inverse. Just this transduction element just converts the non-electrical signal to the electrical signal. So in the sensing element, it just takes the response, and this transduction element, it just converts non-electrical signal to the electrical signal. Some more problems are there. Artifacts. What is artifacts? That is just a noise. Okay, it is a component or a variable. It's observed while doing an experiment, which is not naturally present. Okay, this random noise generated in measuring instrument, electrical interface, crosstalk, and all other unwanted variation in a signal are considered as non artifacts. That is, if we are measuring in the measuring instrument also, there will be some noise generated or some other issues will be there. So due to this, there will be some noise generated. This is called artifacts. Then energy limitation. The many physiological measurement techniques that a certain amount of energy be applied to the living system in order to obtain a measurement. So for the measurement, we need some external energy. So for example, the resistance measurement requires the flow of electric currents through the tissue. That is, if we want to measure the resistance, mean we have to allow some current to flow through the tissue or blood being measured. Okay, so this will, due to this also, there will be some transducers generate some small amount of heat due to this current flow. There will be some small amount of current generated due to this current flow. It also causes the recording of the value. Then safety consideration, that is a method employed in measuring variables in a living human. Subjects must in no way endanger the life or normal functioning of the subject. Okay, just safety consideration. While we are doing all these measurements, if it affect us, means it is a problem, right? So we are doing this without affecting the patient. That is the safety consideration. Recent emphasis on hospital safety requires extra caution must be taken in the design of any measurement system to protect the patient. Just safety of the patient. Okay, these all comes under this recording problems. Next, just we are going to see a simple topic, man instrument system. That is a man as an instrument. So if we consider ourselves as an instrument, what are the things under us? So the components of man instrument system, there will be a subject, a stimulus, a transducer, signal conditioning circuit, display devices, recording, data processing and transmission equipment. So here this is the patient, transducer, then to the signal conditioning circuit, display, recording, data processing and transmission, feedback will given as a similar to the patient. Okay, have a look on this subject. Subject is a human being on whom the measurements are made. It constitutes a biopotential and a living organism. Some of the biopotentials are electrocardiogram, electromyogram, electroencephalogram, electroretinogram. Electroretinogram, what is that? Just to measure the variation in eye. Okay, electroretinogram. Stimulus. Have you understand about the subject? It's just a human beings or to whom we are just making a measurement. It's just a patient. Okay, assume it's just a patient. Then stimulus. What is stimulus? In many measurements, the response to some of the external stimulus is required. The stimulus may be a visual, flash of light, or some tone, tactiles, or direct electrical stimulation of some of the nervous system. Just we are giving some stimulus to the particular cell or something. Okay, cell or tissues. Transducer. It is defined as capable of converting one of the energy to other. It sends the biopotential, converts to the electrical signal. For example, the thermistor converts temperature to the electrical signal. Strain gauge produce the electrical signal by measuring the pressure. Okay, then signal conditioning circuit. Biomedical signals comes from transducer transferred to the signal condition. So the signals that is comes from this transducer is transferred to the signal conditional circuit. Just conditioning the circuit, it amplifies the given signal. It amplifies the given signal some extent, then process the signal by removing the noise and measure signal parameter. In the signal conditioning circuit, that is the biosignal from the transducer is transferred to this signal conditioning circuit. So this signal conditioning circuit amplify the given signal and process the signal and remove the unwanted noises present in the signal, and also it will measure the various signal parameter. Finally, it will transfer the measured parameters to either display or memory for future purpose. Then the display device, that is the output of signal conditioning circuit just for the display of this. Okay. Next, recording, data processing and transmission. It is often necessary to record the measured information for the recording of the data and transmission. Okay. Then control feedback device. It is necessary or desired to have automatic control of stimulus, transducer or any other parts of an instrument system. A control system. So just control feedback device. Just we need control over the entire system. That's all comes under this control feedback system. This system, transducer, stimulus, signal conditioning circuit, display, control, feedback, recording, data processing, and transmission, usually consists of a feedback loop in which part of the output from the signal conditioning or display equipment is used to control the operation of the system in some way, just giving the feedback as a stimulus. Okay. For the purpose, we are using this control feedback device. These all comes under the recording problems and measurement with the two electrodes. Thank you, dear students. Hope you all understand this topic.